Stem cells and totipotency. In multicellular organisms, cells are specialised to perform specific functions. The process by which each cell develops into a specialised structure suited to the role that it will carry out is known as cell differentiation. Single-celled organisms perform all essential life functions inside the boundaries of a single cell. Although they perform all functions adequately, they cannot be totally efficient at all of them, because each function requires a different type of cellular structure, enzyme, and other proteins. One activity may be best carried out by a long, thin cell, while another might be suited to a spherically shaped cell. No one cell can provide the best conditions for all functions. The cells of multicellular organisms are each adapted in different ways to perform a particular role. In early development, an organism is made up of a tiny ball of cells, all of which are identical. As it matures, each cell takes on its own individual characteristics that adapt it to the function that it will perform when it's mature. All cells in an organism, such as a human, are derived from a mitotic cell division of the fertilized egg cell, or zygote. It follows that they all contain exactly the same genes. Every cell is therefore capable of making everything that the body can produce. A cell in the lining of the small intestine has the gene coding for insulin, just as a beta cell of the pancreas has the gene coding for maltase. So why do the cells in the small intestine produce maltase rather than insulin, and beta cells of the pancreas produce insulin rather than maltase? The answer is that although all cells contain all genes, only certain genes are expressed in any one cell at any one time. Some genes are permanently expressed in all cells. For example, the genes that code for essential chemicals such as the enzymes involved in respiration are expressed in all cells. Other genes permanently switched on in all cells include those coding for enzymes and other proteins involved in essential processes like transcription, translation, membrane synthesis, ribosomes, and tRNA synthesis. Other genes are permanently not expressed. For example, insulin in cells including the lining of the small intestine. Further genes are switched on and off as they are needed. Differentiated cells differ from each other often visibly too. This is mainly because they produce different proteins. The proteins that a cell produces are coded for by the genes it possesses. Totipotency. An organism develops from a single fertilized cell. A fertilized egg clearly has the ability to give rise to all types of cell. Cells, such as fertilized eggs, which can mature into any body cells, are called totipotent cells. The early cells that are derived from the fertilized eggs are also totipotent. These later differentiate and specialize into a particular function. For example, mesophyll cells become specialized for photosynthesis, and muscle cells become specialized for contraction. This is because, during the process of cell specialization, only some of the genes are expressed. This means that only part of the DNA of a cell is translated into protein. The cell therefore only makes those proteins that it requires to carry out its specialized function. These proteins include those required for essential processes like respiration and membrane synthesis. Although it is still capable of making all the other proteins, these are not needed, and so it would be wasteful to make them. In order to conserve energy and resources, a variety of stimuli, which are called controlling factors, ensure the genes for these other proteins are not expressed. The way in which these genes are prevented from expressing themselves includes preventing transcription, and so preventing the production of mRNA, or preventing translation. Stem cells. If specialized cells still retain all the genes of the organism, can they still develop into other cells? The answer depends. There are no hard and fast rules. Xylem vessels, which transport water in plants, and red blood cells, which carry oxygen in animals, are so specialized they lose their nuclei. As the nucleus contains the genes, then these cells clearly cannot develop into other cells. In fact, specialization is irreversible in most animal cells. Once cells have matured and specialized, they can no longer develop into other cells. In mature mammals, only a few cells retain the ability to differentiate into other cells. These are called stem cells. Stem cells are undifferentiated dividing cells that occur in adult animals' tissues and need to be constantly replaced. They therefore have the ability to divide to form an identical copy of themselves in a process called self-renewal. Stem cells originate from various sources in mammals. Embryonic stem cells come from embryos. Umbilical cord stem cells are derived from umbilical cord blood and are similar to adult stem cells. Placental stem cells are found in the placenta and develop into a specific type of cells. And adult stem cells. These are found in body tissues of the fetus through to an adult. They are specific to a particular tissue or organ within which they produce the cells to maintain and repair tissues throughout an organism's life. Types of stem cells. There are a number of different stem cells which are classified according to their ability to differentiate. Totipotent stem cells are found in the early embryo and can differentiate into any type of cell. Since all body cells are formed from a zygote, it follows that zygote is totipotent. Pluripotent stem cells are found in embryos and can differentiate into almost any type of cell. Examples of pluripotent stem cells are embryonic stem cells and fetal stem cells. Multipotent stem cells are found in adults and can differentiate into a limited number of specialized cells. They usually develop into cells of a particular type. For example, stem cells in the bone marrow can produce any type of cell, a blood cell. Unipotent stem cells can only differentiate into a single type of cell. They are derived from multipotent stem cells and are made of adult tissue.